Hi, I'm Aubrie, and this is Io, voted Best Personality in the Home Office, and Callisto, voted Most Likely to Eat Your Spacecraft Experiment. I'm the newest member of the Spacecraft production team at Kennedy Space Center, specializing in microbiology. Now, one thing you'll notice about Florida is how green it is and just how much it rains. This is an important system in that the water that comes down into the roots is pulled up through the plant, dropping off electrons at the electron transport chain of the chloroplast, ultimately procuring carbon dioxide from the atmosphere and turning this into sugars. The sugars are moved throughout the plant to carry out metabolism and respiration. They can also be exchanged at the roots with the important members of the microbiome, specifically rhizobacteria and mycorrhizal fungi. These microbes provide nitrogen, phosphorus, and potassium to the plant, as well as minerals. Now, the important question for the next 10 years is how we're going to use the complexity of this system and better understand it so that we can apply it to the closed environment, or the biological life support systems we'll be using aboard Gateway, and a lunar outpost that will revolve around the moon. We'll use the lessons learned from the International Space Station to make a robust and resilient life support system, harnessing the microbes, the plants, purifying water, and our waste streams. To follow up with this, we'll have experts from our Kennedy Space Center team provide you with a little bit of insight on what we expect for the microbiological future.
To this end, microbiologists will continue to push the envelope when it comes to monitoring the microbiome of the closed and built environment, using cutting-edge sequencing and bioinformatics analysis to develop a comprehensive understanding of microbial diversity, dynamics, and adaptation to the space environment.
We will have developed and tested microbial biocontrol approaches to mitigate potential pathogens and biofilm formation. We will have developed a balanced spaceflight diet from our comprehensive analysis of the astronaut microbiome, and we will have characterized an array of crops that will provide safe human dietary supplementation in spaceflight.
We will have developed space-tolerant plant microbiome systems to promote plant growth and prevent plant disease. This was achieved by better understanding plant disease responses under altered gravity conditions and further investigating seed biopriming to promote plant growth.
We will have developed non-destructive plant health monitoring technologies as well as microgravity-independent water and nutrient delivery systems. These capabilities will enable crop growth demonstrations on lunar landers and habitats, and the supply of pick-and-eat crops to crew food systems on Mars transit missions. We will have deployed a space crop production system on Gateway to study the effects of deep space radiation and microgravity on plant growth, demonstrate automated crop production systems for Mars transit, and evaluate food safety during long-duration missions.
Now let's look at a hypothetical scenario aboard a Gateway mission. Here, the Mooncube plant habitat has arrived at Gateway 1 year before the crew on Artemis 7. The Mooncube is activated 2 weeks before crew arrival on Artemis 8.
Gateway Mooncube Logbook Fed 2228, 2029. CO2 and O2 were added for growth, atmospheric pressure altered. Volatile organic carbon is monitored in real time. Ethylene brought under control prior to startup. Microbial inoculum applied to 50% of seeds via watering system at startup. Monitoring, plant health and microbiome via hyperspectral sensor. Monitoring, plant microbe genome interaction via MoonGen sequencing platform. A hypothetical next-gen sequencing technology. Observation, microbial treatment growth is out of this world. Oh no. Off-nominal watering event. Detected by real-time quantitative monitoring of plant health via AI machine learning system. Resolution, automatic reduction in watering. Resolution, re-inoculation with microbial culture after re-establishing optimal hydration. Confirmation, plant and microbiome return to hyperspectral and genomic baselines. Results, crop saved along with 93% of yield. All microbial treatments survive with no human plant pathogens detected. Outcome, fresh salad tonight.
I hope you've enjoyed our discussion introduction. We know that before we go to Gateway, we need to better understand what good looks like. We will do this harnessing the power of ground control studies, ISS validations, and outstanding researchers like yourselves in collaboration with NASA and its partners. So please let us know, what have we not yet addressed?